Good morning, invertebrate enthusiasts. Today we're going to be talking about the mollusks. We're going to end our examination of mollusk evolution and phylogenetics with two groups, the bivalves, a very diverse group and extremely important, particularly in freshwater environments, and the scaphopods, much less diverse and covered for completeness. So the scaphopods and the bivalves used to be grouped into this, or have been hypothesized to be linked in this group, the diasoma. And you can see, according to our cocked phylogeny from Nature of several years ago, any grouping, the diasoma grouping, if we were to hypothesize that, that's not a monophyletic group because it would ignore the gastropods. So the conchiferans supported a monophyletic group, but the diasoma is a Trebiani group, not one that's supported. We used to think that the scaphopods, or the tusk shells, and the bivalves were joined by the apomorphies of having a mantle and gills, mantle cavity being developed laterally, the valves that are joined dorsally and free ventrally, and a foot adapted for digging. And we don't. So the first group we're going to talk about are the bivalve mollusks, so about 8,000 species, so not as diverse as the gastropods, but particularly, as I said, diverse in freshwater environments. So the apomorphies for the group include this bivalve shell with a hinge that's similar or nominally similar to the brachiopod that we saw earlier, but you remember the brachiopods and the bivalves different in terms of dorsal or ventrally which way the bivalve articulates. They have adductor muscles for opening and closing the shell. No radula, so a big apomorphy that we've seen changed throughout the groups of mollusks throughout the evolutionary, all the different molluscan classes. The radula is completely lost here, so a very derived condition where that ancestral rasping herbivory is lost entirely, and these are entirely filter feeders or suspension feeders. They have siphons for inhalant and exhalant water, and some of them can be incredibly long-lived, upwards of 150 years. So bilateral, so compressed laterally. There's a dorsal hinge up here, and then a large space in the center of the shell that protects the gill lamella. So large gills that are accomplishing both respiration and feeding. No head. A big foot, you'll see several different adaptations of that foot as we go along in terms of how it's used as a pedal sinus to help move and protect the shell itself and the animal inside the shell by driving it into the substrate at the bottom of the water column, and also for secreting Bycel threads that help attach the mollusk to and let it withstand incredible forces, shoreline forces. So each valve or each side of the shell has a dorsal peak up here called an umbo that rises above. So the hinge here and the umbo would be up here, and that's the oldest part of the shell. And then taking a look, and we've kind of rotated the mollusk, so we're looking at it in. So if this is the hinge up here, we're watching it hang down with the mantle cavity in the center, mantle skirts, and then these folds of the mantle down here that aren't included in that simplified drawing that I've added them in. So they're essentially the three folds that are hanging down on either side of the mantle, either side of the valve. Now these folds are incredibly intricate in what they can perform. They are clearly what's secreting the shell. So if you take a look here, we've talked at various groups about the nacreous, the prismatic, and the periostracum layer. So different proteinaceous and different kind of laying down of different elemental structure to both protect and refract light. And then, so on the three edges, we've got the inner layer, the middle layer, and this outer layer. So it's the outer layer and this fold here that's secreting the shell. And that's kind of neat. So this is your text drawing this Brusca text here. Now, if we switch back in time to the really analogous drawing that's in the Rupert, Fox, and Barnes text, and you'll see this fold, this groove that's called the periostracal groove in between the middle and the outer fold. So the outer fold secreting the shell, the periostracal groove, that's essentially what's secreting that. And some of you will be familiar with the fact that if there's grains of sand or other bits of obstruction that get stuck in this periostracal fold, in that extrapallial space, that's where bits of nacreous layer are added onto that obstruction, and that eventually is what gives rise to pearls. So the two valves articulate along a dorsal hinge. There's ligaments up at the top here that are proteinaceous, made up of a protein called conchiolin. There's then two sets of adductor muscles. There's fast twitch and a catch muscle. So you can see that the outer ligament coiled up, and then the adductor muscle joining the two valves across. So when the valves are closed by the adductor muscle, that outer hinge ligament is stretched and the inner ligament is compressed, and then it releases, and that allows the feeding. The foot, which we saw earlier, I mentioned that it could be used as a pedal sinus. And as the diagram is kind of looping across from A through D at the bottom here, showing the inhalant and exhalant valves of the water when it's above the surface, and then when those siphons can be extended and pushed back up to the surface as the foot itself draws the shell down and protects the shell underneath the water. So there's circular pedal muscles down here that contract and send the foot out and underneath. So that's the arrow down here. And then there's further contraction along these central muscles that essentially pinch off and cause the foot to bulge out. That's the pedal sinus, and the arrow's going out. So it's now essentially acting as an anchor. Then the retractor muscles and the adductor muscles pull, once that's anchored down here, it pulls the shell down underneath the substrate.
It's tough being a clam. They have no brains. They're popular in chowder. They won't set any speed records. But they do have a foot. Well, a foot that looks like a tongue, but sometimes that's all you need. Along the shallow coastline of the Atlantic, these surf clams spend their days filter feeding for plankton. Some hide under the sand, only a siphon peeking out to feed. They may have no brain, but they're not stupid. Staying under the sand is a good survival strategy. To counteract this, cow-nosed rays stir up the loose sand to expose the clams, then crush them with their powerful jaws. Other hunters, like this oyster catcher, have a different method to get a half-shell platter. They slice the muscles on this jackknife clam and pop open the shell. They don't have eyes per se, but clams do have special photoreceptor cells. A sudden change in the light means something bigger and hungrier is above them. When they sense danger, they dig down. The fastest clams can dig an inch per second. Is it enough to keep this clam alive? Success. That's one clam that's as happy as, well, you get the idea.
Lordy. So, laterally compressed foot that can act as a pedal sinus. You saw that action in the video there of it kind of sending out and pulling it down, down, down. The other amazing thing that the foot can do is produce these Bycel threads. So secretions, they're actually extended out that act as a holdfast and help adhere the clam or the bivalve mollusk and under some extraordinary pounding pressures of the surf upon the shore.
The water deepens. liquid battle for living space begins. It looks as if the mussels are sticking out their tongues, but in fact they're feet, which secrete thin white threads of gluey fibre. In the aftermath of the storm, they use these Bycel threads to cement themselves back to the rock and to one another. By constantly renewing their foothold, the mussels can hang on to their precious bit of rock in the surf for as long as 50 years.
Some amazing footage there. Not such amazing music, but amazing footage of these threads of the foot reaching out and secreting threads and pulling back in. So these, from a biomimetic perspective, these threads are incredibly interesting. So each byssus is a bundle of threads tipped with little adhesive plaques that you're seeing here. And the threads are then joined at the stem, which inserts at the base of the foot. So they send out large kind of a splay of attachment points. And the mussel can actually, as it resets these threads as they're damaged through time, or as the wave action or repositions the mollusk in the substrate, in the rocks where it's trying to adhere, they can reset the Bycel tension and replace these plaques so that their position is laid out in a direct response by the mollusk, by the bivalve, to the degree of force that's hitting it. So 12 Bycel retractor muscles, 6 on each valve. So the angle there, if there's stationary forces, they're splayed out. If there's a lift or a large kind of return force, they retract in almost like a tent. And then if there's extreme drag, they're set out against that drag. So it's kind of the placement is dependent on the flow rate experienced by the mussel, which is a pretty fantastic adaptation that these relatively simple, certainly reduced, no head, but there's evident response to the environment here that is of great interest to all sorts of, from a bioproduct or a biomimetic perspective. So there's iron bridges that are being formed, and there's some papers in PNAS here from a few years ago that are really interested in figuring out how the Bycel threads are laid out and how they're laid out in response without a high degree of cephalization, how they're laid out in response to the environment, and then how they're repaired and where that degree of coordination is coming from. So if you're interested in making money from mollusks, from bivalves, working with Bycel threads and the chemistry of Bycel thread attachment is maybe an area that you'd be interested in pursuing. So the gills, remember we mentioned the bivalves, these have lost the radula. So all the feeding is happening via the gills, the filtering action on the gills that we'll talk about. And there's 3 different kind of strategies with the gills. And those strategies, A, B, C, protobranchs, lamellabranchs, and septobranchs, are related to an increasing in the surface area, the gills size. SA is equal to surface area, and that increases the available area for trapping particulate matter. So we've got protobranchs where there's one, so the foot, and then 2 little stubby bits of gills here. And then lamellabranchs where there's an intra-lamellar junction between, so essentially allowing for an increase in the folding. So instead of just 2 little tinea hanging down, they can increase the amounts of surface area. And then the septobranch, which we'll talk about in a second, has a very reduced gill area because it's actually, strangely enough, predatory. So the lamellabranchs, this connective tissue here, between the ascending and the descending filament. That's the intralamellar junction between these folded halves that allows them to be folded out and not flopping down on top of each other. So this figure 13.29 from your textbook shows the direction of the currents. There's a ciliary current here across the folds of the gills, and that's counter or at right angles to the inhalant and exhalant flow of the water. So the in coming in and across and out. So we've got the ciliary action whispering and picking up food items that are being trapped by the gills, and then the water being brought in and out from left to right. So septibranchs are a raptoral or a carnivorous mollusk that creates suction in its mantle cavity that allows it to reach out and suck in different prey items. But the majority of the mollusks in the world, of those 8,000 species, are filter or suspension feeders. So you can see here the food groove. This scanning electron micrograph is the food groove along the edge of the gills. And so we've got two different lamella of the palps here. And then the ciliary action feeds in to the mouth in the center. Now you remember when we talked about the simplified ancestral mollusk, we went through some of the apomorphies from the group, and so you've seen this slide before several weeks ago that looked at the ciliary sorting fields here, and the typhocyle, the ciliated ridges, and then this crystalline style that draws in the protostyle, the rotating mucus food string. So the gills are acting as a feeding surface and also as a respiratory surface. They're trapped within those protective valves, and bivalves have several different respiratory pigments, hemocyanin, but also hemoglobin, and the mantle skirt in some cases can also respire. So several of the different respiratory pigments that we've seen within the invertebrates as we've traveled along, the hemoglobin, so it's the most common molecule, but we've also seen hemocyanin and hemithrin. Hemithrin is looks like the blood would look pink and violet when oxygenated or clear when it's not, and hemocyanin is the blue in color. And I think one of the things that you might want to do as you're annotating your Dunn-et-al. Phylogeny, which you see here, this is your Dunn phylogeny. It's just kind of flipped and rotated. And what I've mapped on are the occurrences of hemoglobin, which we've done together before, but now hemocyanin and hemithrin. So what we see here with the evolution of respiratory pigments is multiple origins and multiple losses. So we've got with the occurrence of hemithrins, according to transcriptomics, even amongst some of the lineages without circulatory systems, like cnidarians, sponges, and ctenophores. So where are we going to plot the occurrence of these traits, of the trait of different respiratory pigments? This is the kind, as you've seen, the kind of question that I might ask you on the exam is when presented with the phylogeny, and then the distribution or characteristic characters or character states like presence or absence of different respiratory pigments, to map those onto the phylogeny and to describe for me or to make an argument for others about why you would feel that a more parsimonious or a less parsimonious explanation of the trait occurrence would be supported. So there is some, as I mentioned, some circulation that happens in respiration that happens in the mantle skirt. Fairly simple digestive system with the intestine and the blood acting as a hydrostat. The nervous system is certainly less cephalized, and they're sessile, or mostly sessile. But the pallial folds, again, so secreting the shell, the source of your pearls, if you want to think of them that way. But there's a whole bunch of, in these different pallial tentacles, the mantle, or the pallial folds of the mantle, have extreme adaptations here that include ciliary receptor cells that can be mechano or chemoreceptors. There's also individual little pallial eyes, or photoreceptors, that the mantle secretes or creates, that aren't image-forming eyes per se, but can detect shadows and allow the mollusk, to the bivalve, to be negatively phototactic.
The scallops are also filter feeders. They live on the surface and not only have good eyes, but a quite surprising way of getting around.
So sessile, or mostly sessile. And we've seen some mollusks before. This is an example here. We saw the disco clam flashing of these paleo folds. So that flashing tentacles, that's the clam, that's an edge. Remember, we talked about that, these highly reflective silica spheres, about whether or not this was what kind of communication. Was it a communication between species or within a species? And we agreed, we hypothesized as a group, that this was a signal, an honest signal of toxicity, warning potential predators like that pounding mantis shrimp, that the disco clam also contains this mucus, acidic mucus, and so the reflective pallial folds, essentially giving an honest warning that don't mess with me, or I will spew acidic mucus on you. And all of these, again, adaptations of the mantle, photoreception, production of mucus, the retention of these reflective silica spheres. One of the narratives that we're developing as we go through all of our different molluscan examples is how plastic all of the different things that the mantle can do. Now, most of you have taken 1070, and you remember in 1070 we also talked about adaptations of the paliofold as a lure, in terms of bringing in necessary other vertebrate species that are necessary for freshwater bivalves for the dispersal of this adapted villager larva, the Glaucodium larva. So this is this snapjaw that needs to attach right to the gill folds. It's going to use the gill folds of species like largemouth bass to get out and away. So this fish, this darter fish here, again being an adaptation of the mantle, producing the shell, making pearls, making a lure, dense, shining like a disco ball, producing acidic mucus, the mantle, it does all of the things.
In the streams of Missouri lives the Lampsilis mussel, a simple animal with an extraordinary life cycle. To reach adulthood, its young must spend part of their lives inside a fish, the largemouth bass. To get there, the mussels must make physical contact, a difficult task, as mussels don't swim. But the bass has a weakness. It's a voracious predator of small fish, particularly darters. Even the slightest wriggle of a darter's tail will attract bass. Believe it or not, the fish on the mussel is an imitation, a perfect replica that will lure bass within striking range. The mussel can somehow sense approaching fish and wriggles its lure faster to entice them. If it gets the twitching just right, the remarkable likeness should do the rest. On impact, the mussel squirts its young into the bass's mouth. These snap shut on the gills, like spring-loaded traps. Here they stay, drawing blood from the fish, until several weeks later they drop off as tiny, fully formed mussels.
So small fish might be damaged by this, could die. Big fish, no real effect, no real negative effect. Passengers.
Also a favorite prey of the bass are these striped shiners, and some mussels mimic them. Considering mussels are blind and have never seen a shiner, the likeness is incredible.
So incredible adaptations of the mantle. These near perfect species replicates of the hanging mantle fold that's primarily secreting the shell, and without any photoreceptor or eye or cephalized head or brain, the fold appears to mimic exact species of fish. Crazy. So you've seen this before, of course, in 1070. So you remember this practice for the midterm, looking at which statement about this tree would be true, that how many times has this different trait evolved or not. And we know more things, we're learning more and more things about these relationships, that while there are some specialized species of mussel for particular species of fish, there are also some generalists. But on average, if you look at species of mussel compared to species of fish, this tangogram that was recently published is illustrating that many of these relationships are one-to-one, so very specialized. So a high diversity of fish and a high diversity of mussels, freshwater fish, freshwater stream mussels, characterize eastern, particularly
How many
Eastern
different
North America.
species of mussel can you see here?
Is this it?
Oh,
the question.
Oh, there you go.
So Jimmy got there first. Nobody knows.
I just found it quicker than Phil.
It had to be the last one of this show.
Well done, you're absolutely right.
It's the last one we have.
Yeah, the fact is, it's almost impossible to identify
Even
species.
belgos themselves.
Yes.
Impossible to do or impossible to care?
Well,
Do you think they just go, it's mussels, should we just boil these and eat them? Because they are delicious and times are wasted.
we used to think by size and appearance you could tell which species was which. We now find it seems it's only the genome that tells us species we thought were completely different, we've discovered are the same, and conversely, species we thought were the same turned out to be different.
So which nation are secretly training their citizens to be able to tell what species of mussel? The Albanians have mussel ninjas.
It's almost certainly the Albanians, you're right.
The world needs more mussel ninjas. Thank you, Phil Jupitus. So an extreme diversity of bivalves, particularly in freshwater environments. So we have soft bottom burrowers, and attached epa and unattached epifauna. Some of them are boring, like shipworms, where the valves, the different halves of the shells, are used as drills, and they can originally would have been drilling into clay, but now are in especially long shipping lanes in North America and in Europe in an era of shipping when most ships were made of wood. These were extremely economically disadvantageous bivalves. So this long shipworm here, structurally weakened, and so you can read about, if you want, the ravages of Torito, the rise and fall of shipworms in U.S. history, and the collapse of not just ships but also ports and wharf structures that were based around the structural weakening of the support beams caused by these boring bivalve mollusks. So we've also seen that they can be commensal parasites, often, and some of them are floating around inside of echinoderms, and then in freshwater systems, like we just saw, that there's lures, the loss of a planktonic villager, and the adaptation of this glaucodium larva. In freshwater ecosystems, bivalves have incredibly important roles, that they filter phytoplankton, bacteria, and particulate matter. And in doing so, they alternate, or they affect nutrient dynamics and the bioturbation, the circulation of sediments through the water column. And so the physical presence of bivalves creates habitat primarily and secondarily for other organisms, and then changes the nutrient cycle within the aquatic environment by the filtering of sediments out of the water column, and then the deposition of feces and pseudofeces that can secondarily alter the composition. And this is an incredible feature of Eastern North American freshwater systems, but unfortunately, one of the characteristics of Eastern North America in the past 200 years is that we've dramatically changed the unionid, the burrowing bivalve fauna within Eastern North America, and many of our native species are declining at catastrophic rates. And so this is changing how benthic biomass is being partitioned and laid down, and it's coupled with the fact that there's more and more bivalves that are arriving as invasives or non-endemic species from Europe, like the zebra mussel. So many of the things that are native to here are now like the wavy-shelled, wavy-rayed lamp mussel, which is one of the lure ones that we saw earlier, that is now special concern, ranked by CSIWC, or the Committee on the Status of Endangered Wildlife in Canada. And those many more of the mussel species are kind of ranking farther and farther towards endangered as CSIWC gathers data, or scientists gather data to give to CSIWC to list these species. This is true even in, this is a study from a couple of years ago that looked at the mussel diversity up and down the Grand River watershed upon which we're sitting right now. So the Speed and the Aramosa. The Aramosa flows into the Speed in Guelph. The Speed then downstream of Guelph flows into the Grand, and the Grand River eventually flows into Lake Erie. And what this study was looking at across the length and of the Grand River watershed was the abundance and the diversity of mussels and the size class. So mussel abundance was lower downstream of urban areas, so even just the urban development. So upstream of Guelph, there were more mussels, and that they had many more different size classes. So there were big mussels and small mussels. Remember, mussels tend to be long-lived, and so what this is suggesting is that abundance is beneath cities, as you have an increase in wastewater treatment and stormwater runoff. It's reducing the diversity of species, and also disproportionately affecting the younger ones, the smaller mussels. And this is problematic, and it suggests that there's a big problem coming as those big mollusks that have survived die. And we're also then seeing an increase in the invasive mussels, or the non-endemic mussels. So if you want to know more about some of the effects about endangered and non-native species, there's a 5-minute video here that I would suggest that you go and have a look at it. It features some research from across Eastern North America, but particularly some graduate students and researchers at McGill who've been looking at the invasion of non-native mussel species and how that's affecting freshwater ecosystems in Eastern North America. So finally today, a couple of slides to cap off our view of the diversity of mollusks, the scaphopods, or tusk shells. Scapho, so pod, foot, just like with all the other groups, and scapho from the Greek meaning spade. So they're tusk-shelled, conical shells, open at both ends, and the biggest apomorphy that characterize them are these captaculas. So the captacula is kind of like the Bycel thread, but these thread-like adhesive feeding tentacles that you can see here reaching off and helping embed. So the shell being open with the foot anchoring it here underneath the sediment, and then siphons and water movement poking above. And they live most of their adult lives buried in the sand or mud, with the head end, or where the foot is, pointed downwards. And the mantle tissue there, because of the reduction of the amount of the mussel that is exposed to the water, where the oxygen would be, that there's the mantle tissue here is highly adapted as a respiratory surface. So that's it for the mollusks. One large group, incredibly diverse, enormous changes within the group in some of the principal apomorphies. So what can the mantle do? What can the shell do? What can the foot do? think about those as you're thinking about and coming back to review mollusks. And that's mostly it. We'll dip back into the Spiralia to deal with some of these little tiny groups that we haven't talked about. But next up, we're going to talk about the ecdysozoans. We're going to talk about nematodes and nematophorans and a whole bunch of really fascinating groups there.